Hello, everyone, and welcome now to basically more chapter six. And where in this kind of chapter, we're really going to be talking a lot about, immunologic tolerance and autoimmunity. So, what is, let's say, the overview, the biggest overview when it comes to immunologic tolerance? So, we spoke a lot about how B cells and T cells develop and how they're basically, we try to create an army of B and T cells that are specific to an antigen. Well, we need to be careful there because we want to make sure that these basically B and T cells are actually able to recognize self and non-self. Because if they don't, this is where autoimmunity can come in. Because if they start to recognize our own self proteins as an antigen and will start attacking it, then basically they can technically kill us. And this can cause so many autoimmune diseases that we will be talking a lot about today. So in some way, we need to kind of induce some way, help them to identify between self and non-self, and that's called immunologic tolerance. And there's two different types of tolerances. There's central tolerance and peripheral tolerance, which we will be talking about more in details. But just quickly, central tolerance, what that technically means is that when we're creating the T cells, so during the basically its development in the thymus that we really went into so much details, there basically you're literally looking at T cells that are able to react to self protein and not. So what you do is you take literally this antigen presenting cells, which we'll be talking about them. They're called literally MTACs. They literally have an MHC, which they present self antigens. If the T cell is able to recognize it very high, it's gonna cause them to go into apoptosis. If they don't, and if it's very low, then basically are able to go. So this is called central tolerance. It's basically where in the thymus you start to kind of, let's say, edit the T cells. T cells that have a TCR that are able to bind to cell peptides are killed, while T cells that are actually have TCRs that are not able to bind to cell peptides are released. So that's the first central tolerance. We're going to be talking a lot about this soon. And then you have peripheral tolerance. So from the central tolerance, we always have some basically T cells that are able to recognize self peptides that are basically escaping. You always are going to get that. Nothing is 100% secure. If that was true, then basically, if everything was 100% tight, then we would never have any diseases. So to compensate for that, we have something called peripheral tolerance. So peripheral tolerance is basically more in the periphery. This is basically literally all around your body. You have basically many mechanisms that are able to actually stop these basically these kind of T cells that are able to kind of recognize self antigens. And one example is, for example, the use of Tregs. But we're going to be talking a lot about peripheral tolerance as well. So you always have to remember that central tolerance happens in the thymus during the development of T cells, and where you basically are trying to remove as much as you can any T cell that is able to recognize self. Periphery is more basically when they escape and they're literally in your blood system going around. There is basically when they actually are able to recognize any T cell that is able to let's say bind to self antigens. That's where you can actually play around. So to understand central tolerance, let's go back towards the development of T cells. So this is literally taken from your lectures on T cell development, in where we have your T cell precursor. This technically most probably is your common lymphoid progenitor, your CLIP, and where it's in your basically your thymus, and it starts to go into your basically double negative one, double negative two, and then your double negative two, you start to get your beta chain, and then basically once you're basically in your the double negative three, you have your beta chain. You basically have a pre TCR, so you have basically this invariant T alpha chain, which comes together and gives you this pre TCR. There's then signals in your DN4 to get your full alpha chain, and then you have your full TCR, which then gives you into your double positive. In your double positive, you have basically a T cell that is both CD4 and CD8 positive. It still doesn't know where to go. Does it go to the CD8 pathway, so it becomes more of a cytotoxic T cell, or does it go into more of a T helper cell, a CD4 positive T cell? So it's there and there. It's still trying to kind of, let's say, understand where to go. So these are basically, as I said, you have your T cell precursors. They become into a double positive CD8 and CD4. And of course, these have a full-blown T cell receptor. So the first thing what you do is you do something called positive selection, in where you try to present them with basically an MHC with any random peptide. And this peptide could be anything inside basically this antigen presenting cell. This could be actin, this could be microtubules, whatever. But what it's trying to do technically is it's trying to test, does this T cell receptor able to recognize MHC? Yes or no? So if it's able to do so, if it actually is able to bind to your MHC, then it's able to live. So this is hence why it's called positive selection. So if it's positive, it's able to recognize your MHC, then it lives. If it doesn't, they die. And we call this, they basically die of neglect because they don't have any signaling. Then basically, if they don't have any signaling, they won't survive. So this is your positive selection. And this occurs technically in the cortex of your thymus. So within your thymus, if you guys go back to your lectures, in the thymus, there's literally the medulla and the cortex. This positive selection happens in your cortex, and these antigen presenting cells are literally called thymic cortical epithelial cells. So these antigen presenting cells are called thymic cortical epithelial cells. So literally they're epithelial cells from the thymic cortical zone, which come up and they have both MHC1 and MHC2, and then they go into these double positive cells, T cells, and to see if they're able to recognize it. If they're able to recognize it, they live, as I said. If the double positive cell is able to bind to an MHC1, then it becomes a CD8. If it binds to basically an MHC2, it's a CD4. So this is basically this thymic cortical epithelial cell is able to also push this double positive into CD8 or CD4 as well. So this is where you have your positive selection. But there's a Goldilocks rule. So I don't know if you guys are still, let's say, quite young to know the Goldilocks rule, but it's a very beautiful, let's say, fable, kind of the story that we used to tell, they used to tell me as a kid, and hopefully I will say this to also my kids. So there used to be basically this little girl that went randomly in the forest into a house, and then basically she was trying out everything. She had this case, basically soups. The first soup she took was too hot. She didn't like it. The other one was too cold. She didn't like it. The other one was just right. Well, this is the exact same thing when it comes to the positive selection. What happens, technically? If the T cell here is able to recognize your thymic cortical epithelial cell, perfect, if it's MHC. If it's too strong, this binding, it's too dangerous because you don't want it to be too strong because that could mean it could actually bind to your MHC and be activated just by the MHC. So you don't want it to be strong, so you kill it. If it's too low, you don't want it. It's gonna die because there's no use to it. If it's literally moderate, it's a nice, in between high and low, it's perfect. So then it's the Goldilocks rule. This reaction between the double positive cell and the MHC from your thymic cortical epithelial cells has to be moderate. It cannot be too high, it cannot be too low. It has to be perfectly moderate. Okay, so now from your positive selection, you finally have your either CD4 or CD8 T cells. So one of them. So you have either CD4 or CD8 T cells. So it's one of them. Then basically from the cortex, they move into the thymus, into basically the medulla area. So this is the cortex, this is in the thymus, the cortex area. They move into the medulla. In the medulla, they go to something called a negative selection. So this is a bit different than basically your positive selection, in where you have basically epithelial cells, but from the medulla. So we call these MTECs. These are called medullary thymic epithelial cells. These ones over here, we call them thymic cortical epithelial cells. Both are epithelial cells, but these are coming from the cortical zone, hence thymic cortical. While these ones are coming from the medulla, hence we call them MTECs, medullary thymic epithelial cells. And what happens? These cells are trying to test these basically new CD4 or CD8 T cells. Hey, can these guys actually bind not to the MHC only, because now we know that they bind to the MHC, but do they recognize self-antigens? So what happens within these basically MTECs, they start to actually produce a lot of self-antigens, and they keep presenting them to CD4 and CD8 T cells. If there's basically three things that can happen. If they actually bind very weakly, they do not really kind of are able to recognize that they're very, very weakly, that's perfect. Because that tells you that they're not going to bind to self-antigens. They become mature, naive T cells, and now they can basically leave. If they bind moderately, they are able to recognize it, but not too strong, they become Tregs. These regulatory T cells that are literally dampening or stopping immune responses. And we will be talking a lot about Tregs today. If it's too strong, if they react too strongly with this peptide, it means that they're able to recognize extremely well self-antigens, they die. So hence negative selection. They're literally looking for the ones that are able to not bind or bind a bit, not too much. The ones that bind too strongly are killed off. So that's negative selection. And that's your central tolerance. Your negative selection, we also call it your central tolerance because this is where you're able to filter what T cell goes out and what T cell doesn't. And you're filtering based on if it's able to recognize a self-antigen. And one thing that we see with these MTECs, so these are the MTECs over here, these are your basically medullary thymic epithelial cells. These guys, they have something called IRE. I know, guys, I know IRE, it's basically a very funny name, but use it to remember it. Basically, this IRE basically forms this complex in which literally what it does, it causes the expression of a lot of self-antigens. So IRE is literally its major function is to actually be part of a bigger complex that is literally producing a lot of self-antigens. So these self-antigens are then being able to be put in the MHC to be used as a test if the CD4 or CD8 T cells are able to actually bind to it or not. So and basically what happens again here, IRE basically comes in, forms a complex, which basically goes into your genes and starts the transcription of many basically, a lot of these kind of antigens, which we call them basically these tissue restrictive antigens. These are antigens that are restricted to tissues. This could be basically antigens, for example, of insulin, which are restricted to the pancreas. This could be acetylcholine that are restricted to basically to neuromuscular junctions. This could be, for example, myelin, which are also found in basically your neurons. So these are different types of antigens that are restricted. So IRE creates a complex that increases the production of these tissue-restricted antigens. And these are antigens that you don't really want a T cell response against. So you put them in your MHC, and then you start to see if, for example, this is your immature cell-reactive T cell, if it's able to really bind super strongly, death. If it binds a bit, moderately, it becomes a Treg. If it doesn't bind, perfect, it releases. There has been actually seen in certain cases, and these are mostly kind of, let's say, babies, you see that they actually don't have, don't have IRE. They have some sort of mutation in IRE that doesn't work. So what happens? These people actually get a disease that has multiple different types of autoimmunity. It's not just one autoimmune diseases, it's literally basically almost all of them. Because this central tolerance, this filtering of T cells that are able to bind to self-antigens is gone. And you just have a lot of T cells that can bind to self-antigens. Or you're just literally unleashing a T cell army against your own self-antigens. So this is extremely dangerous, this disease. So now comes your peripheral tolerance. Now finally, basically you have, let's say, the release of your cells. So if you come basically here, so you have your conventional T cells that are leaving because they're basically, they don't bind to anything and they leave. Sometimes there are cells that actually bind strongly to a self-antigen, and these are dangerous, and they don't die. They, for some reason, they just don't die. They are able to kind of evade, and they go into your blood. These are dangerous, and you need to have a certain mechanism that are able to control them, and we call this peripheral T cell tolerance. So there's three types of ways that you can actually control them: through anergy, or through deletion. Okay? So, a normal T cell response, so let's imagine here this is your dendritic cell. Your dendritic cell is able to recognize basically here this T cell basically is able to present a, let's say, a non-self antigen. So let's imagine this is your influenza antigen. It gives you your T cell. As we know, you need three signals. The MHC TCR signal, that's signal number one. Signal number two is the co-stimulatory molecules, which in this case is B7 coming from the dendritic cell and CD28 coming from the T cell. And then basically your third one, which is cytokines. And this causes literally T cells to start proliferating and being activated. In the case in where you have actually a self-antigen, and this self-antigen basically is able to bind very weakly to your T cell, so this T cell is able to recognize this self-antigen weakly, you don't get actually any of this co-stimulation and nothing of this kind of cytokines. So you only have signal one. Well, this actually without signal two and signal three, so the co-stimulation and the cytokines, well, the T cell doesn't do anything. And it goes into basically a state that we call anergy. So that is basically a state that is unresponsive and not functional. So it's able to recognize the self-antigen, but it doesn't do anything. And this is usually basically what happens when you have very little, let's say, when you have very little, basically kind of binding. You don't have much binding. So meaning that the TCR is able to weakly interact with the self-antigen. Then basically, of course, within the anergy, it's either that you have no co-stimulation and nothing, so basically signaling's blocked, or rather than having, so in this case, for example, here, the normal of the co-stimulation is CD28 and B7, you actually have an inhibitory receptor. So your dendritic cell realizes like, hey, this is a self-antigen, and this naive T cell is binding weakly to it, so it's dangerous. It upregulates rather than a B7, it basically upregulates a basically inhibitory receptor. And this inhibitory receptor can actually tell the T cell, hey, do not get activated. And basically you go into anergy. So it's either that there's no co-stimulation and the cell goes to basically anergy, or do you have an expression of an inhibitory receptor? So I'm going to give you an example of two very, very famous inhibitory receptors. One of them is CTLA4. This CTLA4 is a member of the CD28 receptor family. So they're very similar to the CD28 family, and they bind to B7. So basically, the CTLA4, it kind of binds to B7 on basically was just reading something. Yeah, okay, perfect. So Tregs can upregulate this CTLA24, basically, the CTLA4. And basically what happens is that this CTLA4 can basically binds, it's actually a member of the CD28 family, meaning that it binds to the same thing as CD28, which is the B7. And basically what it does is that it competes with your CD28 with the B7. The thing is that CD28 is it's activating. So if CD28 basically matches to CD27, it gives a positive sign. Be like, hey, you get the co-stimulation to start proliferating. But what happens is that CTLA4 gives you a negative. Basically tells it like, nope, you should be going into anergy. So what happens technically is that your T cells start to upregulate CTLA4. Sometimes CTLA4 is found on your Tregs. So what happens is that your Tregs can actually come in and actually literally stop your dendritic cells as well to do anything basically. So either that the CTLA4 is actually found on your naive T cell, or it's actually found on your Tregs, and your Tregs can actually come in, can bind to the B7 dendritic cell, stop it, and then also stop your basically T cell. So that's basically how they're able to induce peripheral tolerance, these inhibitory receptors. Another very famous one is PD1. PD1 is also found on your T cells, and basically this can give a negative, literally a negative co-stimulation. And it's basically PD-1 binds to either PDL1 or PDL2. And this basically can actually, if PDL1, PDL2 binds to PD-1 on your T cell, this has an item, which basically a immunoreceptor tyrosine based inhibitory motif. So literally it's the opposite of an ITAM that we spoke about that's activating. This is inhibitory that literally gives your T cell this negative kind of, let's say, signal to stop basically proliferating. So in anarchy, it's either that there's no co-stimulation or there's inhibitory receptors, either CTLA4 or PD-1. Another way to basically to stop in the peripheral tolerance is Tregs. So Tregs, these are the ones that basically, so if you go back in here, these are the ones that when they bind, these T cells bind moderately to the self-antigen, they become Tregs. Because these are great cells that you can actually go and start scavenging. And they can actually start looking for literally T cells that are able to recognize any of these self-antigens because they themselves recognize them. But they will actually, if they're activated, they can actually stop anything. So how they're able to stop? Well, three ways. Either they upregulate CTLA4 and they literally go into dendritic cells and stop dendritic cells from presenting to naive T cells, these self-antigens, so through the CTLA4, or producing of immunosuppressive cytokines like interleukin-10 and TGF-beta. TGF-beta, all it does, it stops the proliferation of T cells. So you're stopping any T cell that is literally recognizing a self-antigen from proliferating. Interleukin-10 stops any immune response like macrophages or dendritic cells from presenting. So you have the production of these two very inhibitory cytokines. But what also these Tregs can also do is they can actually consume interleukin-2. So as we know, the third signal that T cells need is interleukin-2. So what I can do is actually can literally kind of compete for the interleukin-2 and remove it so that the T cells basically don't take their third signal and they do not actually start to produce more. And the final, basically, way of peripheral tolerance is the deletion completely. And this usually happens when, so if you go back in here, in this case, when a dendritic cell presents a self-antigen, and this cell is actually able to bind to it extremely well, so it has a very high affinity, that high affinity is not normal. And T cells usually, if they have that high affinity, they will literally go into apoptosis. So literally what happens is that the T cell starts to actually increase TNF receptors and also FAST receptors, which basically will bind to the FAST ligand. And this will induce the killing. Literally will induce the killing of your T cells if it's too strong, because in generally you don't want something to bind something too strong because it can be too much activating. Even if it's a non-self antigen, too very strong activation can induce the death. So you want something to be in the middle. And basically mutations in the FAST or FAST ligand, so basically these guys has been shown to cause this literally syndrome called autoimmune lymphoproliferative syndrome, and where your T cells that are able to recognize extremely well the self antigens will start to literally survive so well because they don't have this deletion anymore, and they start to basically produce so many different types of autoimmune diseases. So this is literally what happens here basically with when it comes to your peripheral tolerance. Either it goes to anarchy in a sense that there's no co-stimulation, or there's the inhibitory receptors like CTLA4 or PD-1. There's the suppression of Treg cells through CTLA4, or basically through removing interleukin-2, or by giving other cytokines like TNF-beta or TGF-beta or interleukin-10. Or you have the deletion when you have too strong of a binder. You also have the same thing actually happening with the B cell, basically tolerance. So like your T cells, they're going through tolerance. B cells also go through tolerance in a very, very similar manner, but you have some basically differences. So sometimes when we have during the development of a B cell in the bone marrow, we see that if basically you have a B cell that is able to actually recognize a self-antigen in a high concentrations, or if it's present a lot in the membrane, you basically, this actually increases the activation because now you have avidity. So you have the binding of multiple antigens, which basically push much more the activation. Well, if that actually causes something called to do receptor editing. So what happens is that these B cells that are able to recognize these self antigens at a quite high concentration, they basically will reactivate the RAG1 and RAG2 genes to do again the VJ recombination of its basically kappa light chain. So you create a new B cell receptor. So you're trying to edit again this B cell receptor that is able to bind to self antigens. So that's one thing you're trying to do. Or what happens is that if they basically bind to antigens at low concentrations, you have low avidity. So high avidity is if when it's able to bind to multiple. So it's not this has nothing to do with affinity. This has to do with the amount. So if it's able to bind to multiple of them, that's high avidity. It can go into receptor editing. If they basically bind to antigens basically that have low avidity, so basically very little of them, you basically go into anarchy. So they do not get the costimulation, the CD40 and CD40 ligand that they need. In the case that basically, in this case, they still are binding, even through the B cell receptor editing, they still are binding, and they're binding extremely well, you get the same thing as your T cells. You get deletion. Basically, you start to upregulate these fast ligand receptors and TNF receptors that induce the deletion of this whole B cell because you don't want it. So this is how the B cell self-tolerance occurs. The peripheral, you have the exact same thing. So you have the anergy, meaning that if it's basically binding moderately to self-antigens, they do not get literally the co-stimulation, and they actually down-regulate their own B cell receptor. You get deletion if they are able to recognize extremely well antigens, because they shouldn't, and they go into deletion. Or you have Tregs, or you have something else called T follicular regulatory cells. These are, so if you remember the T follicular helper cells, these are T follicular regulatory. These are more specialized T cells for your basically B cells that are able to stop it. They are able to kind of stop them from proliferating in the periphery. So this is basically all the mechanisms that we are able to keep someone healthy with no autoreactive T cells, no autoreactive antibodies, so hence no autoreactive B cells. That's how we're able. But sometimes these mechanisms don't work exactly 100%. And what you get is you get autoimmunity. And what autoimmunity means is that you have the creation of T cells that are able to recognize self-antigens, or B cells that are able to recognize self-antigens and they produce autoantibodies. So antibodies that are able to bind to us, hence the auto is self-antibodies. This is an autoimmunity. When it becomes an autoimmune disease is only when you start to actually see symptoms. So autoimmunity is when you have T cells or self-reacting T cells or autoantibodies. When it becomes a disease, the autoimmune disease, is when you start having literally the manifestation of symptoms. So please bear that in mind. So mechanism of autoimmunity. So autoimmunity for a long time, for decades, has been kind of a dark box, something that we've not been able to understand. Our understanding of autoimmunity has grown a lot, but it's still not optimal. We still actually there's a lot of things we do not understand about autoimmunity because it's an extremely complicated process, honestly, which is super interesting in my opinion. But the first experiments where they really started to understand, like, hey, what autoimmune diseases is when they took literally two genetically identical animals. So they literally took two genetically identical mice. These guys have the exact genome. So they're literally identical. They're literally identical copies of each other. What they did is they took a tissue, they grounded up that tissue, and they injected it. So from mouse 1, they took a tissue, they grounded it, they injected it in mouse 2, which is genetically identical, and they started to see that this mouse started to get sick. They started to get autoimmune. So they started to see that literally you could actually induce an adaptive immune response by giving self antigens. And this is what literally autoimmunity was. But then of course some weird things started to happen. They started to realize that people that had certain infections were developing actually autoimmunity. And specifically, for example, cytomegalovirus, Epstein viruses, different other types of herpes viruses, they started to realize that these people, when they got infected with these viruses, they developed autoimmunity suddenly. And then with more careful molecular inspections, they started to see that, hey, these antigens, these epitopes, these kind of viral antigens look very weirdly very similar to self antigens. So the T cells were actually becoming activated towards these T cells, sorry, these viral proteins or viral antigens, but they resembled so much the self antigens that they started to kill the self antigens as well. So they started to realize, okay, so either you have a self antigen that's exactly the same, or something that resembles a self antigen that can cause autoimmunity. So when it comes to classification of these autoimmune diseases, it's still uncertain the science of classifying it, but they started to see two different patterns. They started to see autoimmune diseases that were more, let's say, organ specific, which we call them literally organ specific autoimmune diseases, meaning that they will only hit one organ. This is an example for this is type 1 diabetes. It only hits your pancreas. For example, basically Graves' disease, which only hits basically your thyroid. Basically myasthenia gravis, which only hits your basically neuromuscular junctions, basically. Or then you have autoimmune diseases that literally are hitting every single tissue or multiple tissues of your body. This is scleroderma, lupus, rheumatoid arthritis. These are able to basically affect literally tissues that are found everywhere. Hence, they're more, let's say, systemic. We call these systemic autoimmune diseases. And the thing is that from both of these, they literally, most of them become chronic. Not always. An example is Hashimoto. Hashimoto's disease actually at one point it basically stops, but the majority of them, they become chronic because these antigens that they're literally now, these self antigens that they're recognizing, they're not cleared because this is your body. This tissue is keeps basically forming again and again and again. So you have a chronic activation of your immune system that hence why your autoimmune diseases are literally for life technically because you're not able to remove that antigen. So you're not able to resolve that immune response anymore. So when it comes to the main culprits of autoimmune immune diseases, they're either self-reactive T cells or your autoantibodies. And we'll be talking a lot about different examples about them. So literally one of the first ever kind of say experiments that really kind of told us like, hey, antibodies can be cause of autoimmune diseases was actually these kind of pregnancy tests, and where they saw basically people that have Graves' disease. So people that have Graves' disease are literally people that have an autoinflammatory disease. They have antibodies against basically this thyroid stimulating hormone receptor. So this is a very important receptor, which we'll be talking about later. That's very important for your thyroid. It's extremely important to produce a lot of thyroid hormones for development. So what happens is that we create antibodies that are basically attacking it. So one thing that we started to see that people with these Graves' diseases were passing on these antibodies basically into the placenta, into the baby, because antibodies are able to pass through the placenta. For example, IgA or IgG are able to pass through. They go inside, and suddenly now these babies are able to actually suffer from basically Graves' disease, even though they don't have, let's say technically, they don't have and B cells that are able to actually recognize basically these thyroid stimulating hormone receptors. No, these are coming from basically the mother. So technically, really, the culprit is the antibodies. And if you do something called plasmapheresis, where you remove the maternal antibodies, this basically now is cured. It doesn't have Hashimoto anymore. So this was one of the first experiments that really told us, like, actually antibodies can be a cause. And it can be a cause of multiple different types of autoimmune diseases, which I will be talking about. For example, I'll be talking about Graves' disease and also myasthenia gravis, because they're very important ones. But also T cells was also a very cool experiment. This was in 1989, if I'm mistaken, this experiment. That what they did is they literally took this mouse, they injected it with myelin. So myelin is this protein, is this protein that is found literally on your neurons. It literally helps signals travel faster. So what they did is they injected myelin basically in this mice, and suddenly these mice started to become paralyzed. So this is basically a normal mouse. This is basically the mouse that we injected now with the myelin. You see, it's really paralyzed. It's not able to move. It has these flaccid legs and doesn't able to do anything. And this is basically now this guy actually has which we call basically EAE, basically experimental autoimmune encephalomyelitis. So it's an autoimmune that basically you create an inflammation in your myelin in basically your technically in your basically neurons. So what happens is that you're creating a T cell. So basically, what they started to see that you started to create T cells against myelin. So to actually be like, okay, is this really the case? So they took the T cells from this paralyzed mice, poor thing. They took the T cells and they injected it in a new basically mouse that doesn't have anything, doesn't have the autoimmune disease, and they saw the exact same symptoms. So they started to see that it's the T cells actually can cause this autoimmune disease. So the main culprits are antibodies and T cells. So in this case here, you have T helper 1 cells, but of course, it's not just T helper 1 cells. We actually start to see T helper 2 cells, T helper 7 T cells, and they can give to many different types of autoimmune diseases like vitiligo, pemphigus, and psoriasis. Again, I don't need you to know this, but all I want you to know is that it's not something that is just T helper 1. It can be basically made from many different types of T helper cells. Could be coming from cytotoxic, but also from CD4 T cells as well. But one thing that we also see that is very dangerous within autoimmune diseases, you create this literally self-amplifying loop. And by that I mean, let's take this example. Let's imagine you have this basically B cell that's able to actually bind to self-antigens. So you have this is your one of your cells that suddenly something is happening to it, it dies, and it releases self-antigens. This B cell comes in, it's able to recognize this self antigen, and it gets activated. It comes in, it starts to actually activate. It takes in basically this antigen and presents it to T cells. So what happens? You create a T cell response, but you also create basically B cells that are able to actually bind specifically to the self antigen. Basically, these antibodies start to create a huge immune response. You start to bring many different types of basically other cells like monocytes, macrophages, neutrophils, and these starts to get basically really pissed off and start to release a lot of cytokines and a lot of basically these kind of, let's say, molecules that are literally killing all these basically cells. And the more killing that you do, the more release of other self antigens. So you start to create, to actually bring out other B cells that are able to bind to other self antigens, and you repeat the loop, but now against other self antigens. So now you're literally causing more basically autoimmune diseases, basically autoimmunity, because you started off with one self antigen, now here you have two other self antigens because of this much literally kind of inflammation that you're creating. And this is called epitope spreading. This is literally when you're literally spreading autoantigens, so self antigens, you're literally causing them to spread out more, and you're letting other basically B cells or T cells that are able to recognize them to be activated. Sometimes what you see is that you have these self antigens that are able to be basically actually kind of recognized by either basically by T cells or B cells that are able to recognize it, but they're in so little quantities that it really doesn't do much of an activation. Sometimes what happens is that the auto antibodies that you create, so these guys over here, they will bind to these basically these antigens at a low concentration, And what they do is they increase the basically the amount of it that's actually taken up by your T cells or B cells. And now they become actually more visible. These literally peptides are called cryptic peptides. These are cryptic peptides, meaning cryptic in a sense that they're literally um, kind of in some sort of way hidden peptides. They're hidden. They're very low. The B cell doesn't really uptake it. It's too little. The T cell doesn't really recognize it. It's too little. But now because of autoantibodies, you're increasing the uptake of it because anything when an antibody basically binds to it, it opsonizes it, so it increases the uptake of it. So now you create basically getting these cryptic peptides to be now actually self antigens. So this actually can happen. It can happen because of epitope spreading as well. So these are basically all these are different common autoimmune diseases. So of course, I don't want you to memorize all of these. I'm gonna talk about some of them, but here you have the disease mechanisms and what you get as basically as a consequence out of them. But now let's just quickly talk about, let's say, antibody-based um, autoimmune diseases. And one example is myasthenia gravis. So here you have this neuromuscular junction. So this is basically your muscle, and these are basically your neuron cells. Hence, this is why they call the neuromuscular junction. And when basically you want to move, let's say your muscle, it doesn't move just because it wants to. It's because when a neuron is able to release basically this acetylcholine, so this is a neurotransmitter. This acetylcholine will go into your muscle and will literally bind to these acetylcholine receptors found in your muscle cells. And this causes an influx of sodium that causes the muscle to contract. If there's literally no sodium influx, so these basically acetylcholine receptors are not activated, the muscle is relaxed. So what happens is that some B cells are able to actually recognize these acetylcholine receptors as antigens. So they create antibodies against them. And what happens is that these antibodies bind to the myasthenia gravis and causes them to be internalized and basically degraded. So what happens is that your muscle doesn't have any more acetylcholine receptors, is not basically being activated, there's no sodium influx, and there's no muscle contractions. And you start to get, for example, in this kind of kid, this kid has basically droopy eyes because the muscle is relaxed, is not contracting. This basically can happen, of course, in all around your muscles, around your basically your body, and to the point that this can reach into paralysis. Another example of basically basically the autoimmune diseases that is basically kind of the main mechanism is through autoantibodies is Graves' disease. So what happens in normality? This is your pituitary gland. Your pituitary gland releases basically this thyroid stimulating hormone. And what this basically hormone does, it goes to your thyroid. It binds basically to your thyroid stimulating receptor. So which is basically found here. This basically is able then to allow to basically release this kind of thyroid hormones shown here basically in this kind of blue or green balls. So these thyroid hormones are extremely important because what they do is they basically help us basically in development. They help us to kind of grow and to do a lot of kind of metabolism, and basically they're just good for homeostasis and development for us. So what happens is that these thyroid hormones, they kind of create this negative feedback in a sense that when there's a lot of these thyroid hormones, it goes back to your pituitary gland and it stops the release of TSH because you don't want too much of this thyroid hormone because this could be extremely dangerous. Too much of it, it can be lethal and toxic. What happens basically in people that have Graves' disease is that they actually have a B cell that is actually able to react towards this TSH receptor, this thyroid stimulating hormone receptor, rather than in this case actually then to kind of flag it for degradation. It actually is able to bind to it and further activate it. So literally these receptors are kind of being activated by this antibody. So you have so much production of these thyroid hormones that even that because you have so much of these thyroid hormones, they're going back to your literally to your pituitary gland and stopping TSH. It doesn't matter because these are basically still being activated because they're mimicking the TSH now, and you just keep having a lot of basically thyroid hormones. And what happens? These people have an increased metabolism, that they're extremely skinny. Basically, they actually have a lot of basically kind of cold tolerances. They have, for example, tachycardias because the thyroid also causes some homeostasis within basically your heart pumping. You also get basically these bulged eyes. So people that have these really bulged eyes, this is a big, very symptoms of Graves' disease, actually. So now let's move on from autoantibodies to more T cells. And I guess everyone has mostly heard of basically these are coming literally from type 1 diabetes. So in your pancreas, you have different types of cells. You have basically these alpha cells, which release glucagon. You have basically beta cells that release insulin. And then you have somatostatin, which are basically coming from your delta cells. So as we know, the glucagon helps basically the production of literally what do you call it to release of glucose. Well, insulin is much more of literally taking up glucose, right? So what happens is that you have T cells that are actually able to recognize these beta cells as actually a so basically as an intruder. For some reason, these T cells are able to recognize these self antigens coming from the beta cell. So the cytolytic T cells come in and they destroy completely these basically beta cells that are releasing insulin. So basically, you don't have any more insulin. You're not able to take up the glucose. And this is basically an example of what you see. This is, for example, in your pancreas. These basically, the ones shown here in brown are your beta cells. This is basically what you're supposed to see. Bury a lot of these beta cells around. Suddenly, it's all gone because the cytotoxic T cells have killed them all. And you do not have any more beta cells. You have no more insulin. Another example of basically that you see this is with multiple sclerosis, and these are quite weird because what happens is that in multiple sclerosis you literally have these kind of periods where you're feeling extremely good, but then periods that you're feeling extremely bad. Because what happens is that in the times you're feeling extremely bad, what happens is that something triggers it. We don't know what it is. We really don't know, and something it triggers in a sense where you actually have an inflammation in your basically in your brain. So you have a lot of basically leukocytes, macrophages, neutrophils, monocytes, or mast cells that are literally coming, crossing your blood-brain barrier, coming into basically your brain, and they're literally coming in and basically bringing T cells. The T cells literally come in, they start to recognize your macroglial cells. So these are basically your basically cells basically found around your brain, and they literally start to basically they basically sorry, the microglial cells are actually antigen presenting cells in your brain. So your microglial cells start to present antigens coming literally from your central nervous system, and what it does, it literally activates T cells that are able to recognize it. So you have this T cells that are able to recognize antigens from the CNS. They start to get activated extremely a lot. And what they do is they bring a lot of macrophages, a lot of mast cells, and all they do is start to produce a lot of different, basically inflammatory inflammation, and they actually produce. They bring B cells. B cells start to produce antibodies, and they basically start to activate the complement system, and they start to kill off literally this myelin sheath. So this myelin is this basically protein that's along these neurons, and these really what they do is they extremely help basically your kind of basically your neurotransmitters to go fast. And if you remove them, basically they go slow, and you actually have very delayed responses. So this is basically how, again, your T cells can come up, how the beginning of an inflammation brings your T cells, which then basically creates a huge, basically kind of, let's say, basically immune reaction. So on contrary to here, where you have your cytotoxic T cells that are doing the killing, here you have T helper cells that are literally are orchestrating a huge immune response against your myelin. And you have this actually with literally rheumatoid arthritis. You basically have an unknown trigger that brings all these different types of cells to your synovial membranes. So basically, this could be in your knuckles or in your knees, and they bring T cells. These T cells are able then to basically literally take up, or literally are able to, let's say, kind of get activated by certain autoantigens. They literally will cause your macrophages to literally get very basically to get your basically macrophages to get activated, and these macrophages start to release TNF alpha and interleukin 6, And what these do is these actually go into your fibroblasts, which then make them release basically this matrix metalloproteases and this rank ligands. And what literally these guys do is they literally destroy your osteoclasts. And what the osteoclast does is basically it literally they help you break down bone and create basically your bone. So you're literally really kind of harming basically your synovial fluid, basically your synovial membranes, and you're really kind of removing almost a lot of the bone down there. So you're creating a massive trigger down there. So thank you for listening, and if there's any questions, please let me know. Thank you so much.